more grateful as ever for the fact that you're here and what you do for the museum. I mean, you've heard me say this before, but it's absolutely true that this institution could not operate without its volunteers. So it's great to be able to offer some volunteer enrichment this morning. So Emma's been interested in rocks and fossils since she was a small child. When you see her stand up, you'll realize it's hard to imagine Emma as a small child. She was brought up in New Mexico. I think she, like many of us, drove her parents nuts by filling the house with various objects. And she tells me that she used to walk around largely with her eyes glued to the floor looking for stuff, and in consequence occasionally walked into buildings and other obstacles. She went to McAllister College, private school in Minnesota, and I think originally wanted to do music. Yes. And at some point, and I mean is still a talented singer and musician, at some point had an epiphany. I'm probably due to a lecture from one of her mentors, and decided that really geology was her future in life. And as she's now at Yale, it'll come as no surprise that she graduated summa cum laude. She applied to work in my lab, and I've spent more years than I care to admit trying to work out how fossils form and why they matter. So I'm delighted that Emma is finally going to reveal the secret this morning. All yours.
There we go. So, thank you all for being here. I'm very excited about this, and I'm just going to talk a little bit about what fossils are, kind of give you an introduction, but then also talk about a little bit of my research on fossils and how they form and why they matter. So first of all, if we're going to be talking about fossils, I think we need to define what a fossil is. And the word fossil comes from the Latin fossilis, which just means to be obtained by digging. So that's one interpretation of what a fossil is. But I prefer to think about it as evidence of past life. And so things that commonly are referred to or thought of as fossils are something like this. So you have a leaf or a fern here, a dinosaur, and a trilobite. So these are body fossils, and they very obviously represent past life. But other things, like molecules, can also be evidence of past life. And so a lot of people studying paleoclimate can use various molecules, usually lipids or hydrocarbons, that have been preserved from bacteria often, to look at past climate. So that's one example of a fossil that you may not think of normally. Other things are very, very small, like these pollen grains that I extracted from lake sediments in Glacier National Park. So these are only a few thousand years old, but we have pollen and spores going back to about 400 million years ago. And so, although they're very hard to see with the naked eye, they are also evidence of past life and can be used to reconstruct climate. They can be used to reconstruct vegetation in the case of this pollen. But things that are a little harder to find. But then also, these are dinosaur tracks. So trace fossils are very useful in learning about past behavior. And so these are just dinosaur tracks from Dinosaur State Park a little further north. And those can also be very useful, as everything is, in learning about what the animals did, how they moved, their mechanics, and just general behavior. So burrows are also a great example of a trace fossil. And fossils are all the result of various cycles that occur on Earth. And so everything that is a fossil somehow begins in the biosphere. So these are living organisms themselves. And when they die, they can become remains, and then they can be eaten or returned to the biosphere through scavenging and whatnot. They can then be buried, unburied, reburied, mineralized, and then often what ends up happening is they become a fossil, but then those fossils themselves can be eroded and the nutrients are returned to the biosphere. So everything that we see here in the Peabody, what we have as fossils today, somehow escaped this cycling of nutrients, of organisms. And I'm going to be referring to these cycles often today in my talk, but I'm interested in what actually becomes preserved and then what we find and how we study it. And why are they important? Fossils and fossil deposits let us look into ancient ecosystems, the ancient Earth, and kind of reconstruct how it used to be. And so this is a cartoon of Cambrian ecosystem about about 525 million years ago. And these are the organisms that we know of. But these are the Burgess Shale fossils, or Burgess Shale ecosystem. And this is an exceptional fossil deposit because normally all of these things, like Xenomylcaris, these are soft-bodied arthropods. And the normal fossil record, the majority of what we have here in the Peabody, in fact, are shelly things. So brachiopods, mollusks, clams, snails, all things with shells or bones, if you want to think about an internal skeleton. And the normal fossil record kind of looks like this, and we only get a small glimpse of the ancient ecosystem. But by studying these exceptional fossils, here are some examples, we kind of can reconstruct the ancient world more clearly. We can get a better understanding of evolution and how today's world evolved from these ancient organisms. And so I think in this diagram here, that little guy, number 20, is Pikaia, and that's the earliest example of a chordate, so something with a central nervous line. So that guy right there can kind of be thought of as our ancestor 525 million years ago. So today I'm going to talk a little bit about different types of fossils and how we study them. And then I'll be talking about preservation of land crabs, like this little guy here in the Bahamas, and then finally some fossil leaves from New Caledonia that I collected last year. So first, fossils and paleontological methods. So this is just a list, it's by no means extensive, of common types of fossils in the record. So this here is just an impression of a leaf, and I'll be going through these now. The most common type of fossil is definitely kind of casts and molds, and these are formed, I have an example here at the front, when, say, a shell is buried, and then the sediment around it becomes a rock and liquefies. And that sediment then the rock takes the shape of the fossil inside. And that's what you see here. So this is the negative impression of the external part of the shell. So this is the mold here. And then that shell can be preserved, and that often is the case. But you can also have the shell decay away or dissolve away, and then sediment, different sediment, fills in that mold and becomes the cast. So if you guys came to the front before my talk, or you can look at this after, this is a great example. So there used to be a shell, it was buried, it became a rock, this shell decayed away, but you still have the mold of the shell on one half, and then the cast of different sediment in the other part in 3 dimensions. Another kind of fossil that is quite common is permineralization. And so this is what happens when fluid carrying dissolved ions and dissolved minerals deposits those dissolved ions and minerals in the void spaces. So a great example is permineralized wood. This is an example from the Painted Desert out in Arizona, but we have some wonderful examples here in the Peabody. The curator's choice currently is about fossil permineralized wood from Connecticut, but we also have wonderful examples in the Great Hall of Araucarian trees, or monkey puzzle trees, from the Triassic and Jurassic, kind of to put the dinosaurs in perspective, because vegetation really does rule the landscape. And so here you can see that this is a cross-section of fossil wood that has been impregnated in silica. But the organics remain, and what has happened is that the silica itself is preserved with a 3-dimensional structure, allowing us to look at cellular level detail. So these little holes here are the holes in the vascular tissue of this ancient wood that allowed water to flow through. And by permineralizing it, we can actually go back, look at what that fossil wood was, what species it was, but also kind of understand the ancient vascular system. So there's examples of extinct seed plants from about 300 million years ago that they've actually been able to use wood like this to model how the vascular system of plants evolved. And so permineralized wood has been incredibly important, especially for paleobotany. Related to, but a little bit different, is replacement. And so instead of filling the void spaces around the tissues with mineral, this is the actual one-for-one replacement of the original tissue by something else. So in this case, these are organisms from a 250 million year old reef. So this is a brachiopod, this is a piece of coral. And these used to be built out of calcite, or calcium carbonate. And in this rock, those minerals were replaced again by silica, and we can dissolve away the calcium carbonate rock, and we're left with these fossils. And so in this case, the original tissue is no longer there. It's all gone and replaced by a new mineral. Spectacular examples can be preserved in pyrite, fool's gold. And so a lot of important things out of the Ordovician. So about 500 million years ago, 480 million years ago, are replaced in pyrite, which lets us see spectacular detail on the legs of trilobites instead of just the shells that we normally have. Another example is just rapid cementation. So if you can imagine, in this case, a leaf, but also other arthropods. I have one here in the front that's an ancient worm that basically falls into sediment, and the sediment immediately around the fossil becomes rock very, very quickly, preserving the fossil inside. So this is an example of a Mason Creek concretion. This is from Illinois, and it's about 300 million years old. And what's great about Mason Creek is, at least for the Mason Creek insects, here in the Peabody, we have 76 of the original specimens that were used to designate species names for this locality. So the Peabody has a very deep connection with this kind of fossilization. One of the most common types of preservation for plants and insects is compression or impression fossils, and that's exactly what they sound like. The plant has literally just been compressed in sediment and preserved. This insect, which I have to say, it's a bee, I have to say is my favorite fossil ever because he's just so cute, has just literally been squished and preserved. And so you can see these darker areas do have some original or carbonaceous material left, but the majority of the fossil is gone. It's two-dimensional now. The only difference between compression and impression is whether or not there's organics remaining. So often leaf fossils, you can think about if you walk around here in New Haven and see leaf impressions in cement when that leaf fell into the rock. The leaf is no longer there, but you can see its shape left in the cement. That would be an example of an impression fossil that's kind of man-made, but that's okay. So that's very, very common in things like plants and insects. And one of the most important types, especially for paleobotany or the study of ancient plants, is charcoal. And the difference between charcoal and coal, the coal we use for energy, is coal is a mineral that forms deep in the earth. So it's mostly plant matter that's been buried, subject to temperatures and pressures. And it becomes this mineral over many, many millions of years. Charcoal forms instantaneously in a fire. So charcoal is what happens when plant matter specifically is burned at very high temperatures in the absence of oxygen. And I know that sounds weird because fire needs oxygen to burn, but certain tissues, if they are buried, let's say 10 centimeters under the soil, are subjected to these temperatures, but they survive the process and become charcoal, which is very, very robust. And so these are examples of Cretaceous flowers. These are some of the earliest flowers we know of. And this is kind of helped shape our idea of how the flowering plants, which dominate the world today, evolved during the Cretaceous. And so charcoal has been very, very important. And finally, amber. I know that one of my favorite types of jewelry has amber in it, but amber is also very valuable for studying mostly insects in this case because they're preserved in 3 dimensions. They're in often life positions or dying positions. And once those, basically it forms by trees, mostly flowering plants, but also a few conifers. They produce sap, which I'm sure you've all had battles with because it's terribly, terribly sticky. And what happens is an insect either walks into it, falls into it, flies into it, or is just unlucky enough to be covered in it while sitting on the bark of the tree. That fossil, or that resin, that sap is buried and forms amber. And so amber is one of those things that everyone knows it's useful, and everyone knows it comes from tree sap, but it's very, very hard to study. And so we're not entirely sure of the process between sap and the resulting amber, but we know it's valuable. And so it's proved very useful in studying especially insect evolution, insect histories, primarily since the Cretaceous, since the rise of the angiosperms. So what do you want to do to become a fossil? I've talked about what fossils are, how a few types of fossils form. But if you wanted to become a fossil, what would you do? Have a heart. That's what I was afraid was going to happen. My hair is making a problem. Have a hard body. If you already have a shelly exterior, in the case of these gastropods, or you have bones, you're already a good way there. You're already partially mineralized. So if you become buried, you have a better chance of surviving those cycling processes. You have a better chance of surviving any transport that might occur, or a lot of scavenging is difficult because, like, unless you're a hyena, you probably don't eat the bone, you eat the soft tissues. So the bones can be left. Or in the case of these gastropods, the innards might be eaten, but the shell will remain. And so you have a hard body, whether it's an exterior shell or an interior skeleton, you're already pretty good. And if you get buried very quickly, that would also be useful. So, yeah, if you're sitting on the surface, you have a much better chance of being dissolved away by rainwater or eaten. But if you become buried in the sediment, fewer things can get to you, and you're more likely to survive. So these are two examples. This is another Burgess shale fossil. This is a really interesting arthropod called Opabinia. It has this nice little trunk of a nose, and it had 5 eyes. And so this is one of those stem arthropods that there's nothing really around today that looks like this. So some people refer to this early group of arthropods as experimental. It's in no way inferring creation or intelligent design, but it was early arthropod organization that just didn't work, or didn't survive at least. This is an example of Confucius ornis, which is a feathered dinosaur from China. And in this case, this was buried by ash clouds. And so volcanoes of course, produce a lot of lava, but in explosions like this, they also send out a tremendous amount of ash. And if that ash lands on this poor little dinosaur here, ash is a great way of preserving fossils. So a lot of these spectacular fossils are preserved in ash like this. In this case, there was not ash involved. It was just buried by an underwater debris flow. And so you can imagine he was swimming around, this giant cloud of sediment came down the slope in the ocean, covered him, trapped him, buried him, and that's why we can see him today. So,
what's the size?
This one is about, I think the fossil itself is like 2.5 inches long, and this Confucius ornis, small, medium chicken, you can think with the tail, not including the tail. So a relatively small dinosaur, ancestor of birds. So he's on the line with Archaeopteryx to modern birds. The title is a little misleading on this. You can either die in your friends in a mass mortality event, and in that case it's great because if you die, and let's say you die in this watering hole, things will eat you. But if there's too many of you, they can't eat all of you. So you have a better chance of surviving. The other option is to be one of a very, very large population. So if you're one of a species with millions and millions of representatives, there's a chance that one of those representatives, or millions, will end up becoming a fossil, especially if you have a hard body. So this is an example from Nebraska in which there was a watering hole like this. So this is a modern herd of zebras. And again, it's giant ash cloud buried them as they were dying in this poor watering hole. And so we left with these herds. This is only one of probably 40 or 50 fossil, well, in this case, rhinos that have been preserved in ash in this mass mortality event. So that's die with your friends or be one of many. Dig your own grave. As I mentioned before, burrows are a fantastic trace fossils, but burrows also have the unique habitat in that you've already been buried. You're already in the ground, so instead of having to be buried quickly, if you die in a burrow, you're already underground, you're already protected from a lot of these cycling processes that can remove you from the lithosphere. And so if you are like this little meerkat family, let's say your burrow collapses, it's very, very sad, but you have a better chance of becoming a fossil, so that's pretty good. And live where fossils form. Most fossils are formed kind of at sea level or on flat environments. You can think of a floodplain or shallow, the shallow part of the ocean. So probably up to, I don't know, 1,000 meters, not shallow, 500 meters depth on non-active margins. So if the east coast of the US is a great place to become a fossil right now because there's not a lot of tectonic activity. If you were living on the West Coast, you have a very good chance of being brought down as the plates kind of come together and the oceanic plate is being shoved under the US. So don't live on the West Coast, live on the East Coast. Or live in a nice big lake like this little turtle. Do not live at the top of a mountain. If you're living at the top of a mountain, even if you do fall into a mountainous lake, that mountainous lake and the sediments in there and the rocks it will form will be eroded. That's just the nature of mountains. So live in the floodplain, live in lakes, live in a shallow marine. And finally, you have to make sure that the conditions are right. So this is going back to charcoal. And I said that it formed at high temperatures. But what are those high temperatures? So this is a series of experiments that is done to look at what are the right conditions for our charcoal to be formed from wood. And so they took a block of wood, and up to about 200 degrees Celsius, no matter how deeply they buried it, no matter how much oxygen was exposed, or the wood was exposed to, it didn't do anything. It was basically just a piece of wood. Increase it a little bit, you get some charring around the edges, so that's this brown bit here. It's a little hard to see. Between 220, 250 degrees Celsius, 200 if there's a lot of oxygen. It turns charred, but it's not really going to be preserved. It's still very organic rich. It's still going to decompose. It's still going to decay away. However, charcoal, like those robust flowers that I showed you earlier, can form in a very wide temperature range between 270 and 560 if it's buried a little bit under the sediment, if there's very little oxygen. If it's on the surface, between 200 and 250 degrees. So if you're a little plant living in a forest, there's a forest fire, if it doesn't get too hot, you might become charcoal. Anything over that, this white part here, you're just going to become ash. And if it's really, really hot, you just disappear entirely. So I talked about how you become a fossil. Now, what do we do as paleontologists? How do we study these things? How do we figure out how they're preserved? And the most obvious and the thing that we usually do first is look at them. So obviously, light microscopy is great. So this is just a normal dissecting microscope. And so these are images that I took of this weevil here. And you can see that these color bands on the back of the weevil are formed by these tiny little scales. So there they go. The scale bar is half of a millimeter. And so these are very, very small scales. They're probably, I don't know, less than 1/10 of a millimeter in diameter. But light microscopy really only lets us see some detail, but not anything closer. So we don't know how these scales are producing the color if we just use light microscopy. If we use scanning electron microscopy, so this is the scanning electron microscope over in KGL, and this is where I do a lot of my work. If you crack one of these scales open, you can see very small structures. So I should have put a scale bar in here, but the diameter on this is about 100, not even that. It's about 50 thousandths of a millimeter, 50 microns, 50 micrometers. And these structures in here, this honeycomb-like structure, is the structure in these scales that is actually producing the color. So by using scanning electron microscope, we can see very, very small details. And there are actually fossil examples of these tiny color-producing structures in the fossil record. And so we can instead of just using this modern beetle, we can actually put the fossils in the scanning electron microscope and see these very, very fine details. And this tool here is going to come up later in the talk. We can look at the elemental composition of the fossils. And so by shining or pointing an electron beam on the surface of the sample, we excite the atoms in that sample, which then send off characteristic, both X-rays, but also electrons of a certain energy. And by calculating this energy, we can actually just see what elements are in that fossil, either by looking at a single point. So this is just one small area of a fossil. And so it's composed primarily of oxygen and iron. Ignore this. This is what we have to coat it with so that the electron microscope doesn't fry the sample. A little bit of aluminum. So there's probably, aluminum is a very diagnostic element for clays. And so you can say that the fossil that I was looking at in this case was primarily composed of iron, oxygen, a few clays, so iron oxides. But we can also use this tool to map things. And these are some spectacular tools because you can actually look at the distribution of different certain elements, and therefore the distribution of certain materials or minerals that have preserved this fossil. And so in this example, here, we were looking at the distribution of copper to look at color patterns in the fossil record. So this is a fossil of a chrysomelid beetle, so kind of related to ladybugs, only a little bit. And this fossil here is about 45 million years old, and we put it, we scanned it and looked at the copper distribution, and each one of these little dots, if you looked at the fossil itself, was a little black dot. So that was the color pattern, and that's being preserved in copper. This is an example of mapping a modern beetle, also looking at color patterns. And so if this is just the image that we would get using a scanning electron microscope. It doesn't show the composition at all, it just shows the shape and the size and the morphology, but by looking at the elemental composition, we can see that these black areas have a lot of chlorine and potassium in them. So it can be a very, very valuable tool in looking at distribution of minerals and elements, which gives us an indication of the environmental conditions, but also how it's preserved. So if you looked at a piece of silicified wood, you would get a lot of silica in the map, or you should at least. Experiments are a fantastic tool as well, because we can always look at fossils, but until we really understand the processes that go into forming fossils, we can't necessarily understand the conditions that they need to be formed in. And so experiments can give us at least a little bit of insight into how various types of fossils form. And so this is another example of charcoal. So this is Cretaceous conifer wood. So instead of being silicified, it was produced or preserved in silica, or in charcoal. And these experiments kind of looked at temperature and time at various temperatures to form this. And so the first 250 degrees that they subjected this piece of wood to, if you look at it in this cross-section here, you can't really see any of the cellular detail. If you expose it at 250 degrees for a little bit longer, you start to see some of the cellular level detail, but it's not very clear, and it's still very organic rich. And so even if you had that detail in the fossil record, this would still probably decay away. But at 300 degrees, you suddenly see a very, very similar morphology, similar reflectance. This is charcoal. This here is true charcoal, and this kind of conditions of 300 degrees. There's various types of fire. This would probably be preserved in just like a shrub fire, so not a huge wildfire burning all the trees, but just the ground stuff. That usually is about 300 degrees Celsius, and that has the capability of preserving these plants. If you go too hot, it breaks down completely. We would definitely not preserve that in the fossil record. So by looking at these experiments, we can say, one, that that's about the temperature that's needed to form these fossils. And so if you have a piece of charcoal like this, not only can you say, oh, it was preserved at these temperatures, but you can use that information to say what type of fire produced this. And so you can actually, by doing experiments, not only understand how the fossils formed, but the environmental conditions, the paleoenvironment better just by looking at the fossil. And finally, modern analogs. And instead of going into the lab and trying to recreate environmental conditions, go out into nature. Look at things that are becoming fossils now and try to figure out the processes that go into it. So again, back to silicified wood, if you have a volcano like Mount St. Helens in 1980, especially as Mount St. Helens, it used to be covered, if you look at before pictures, it's covered with a lot of snow. And that eruption of the volcano melted the snow and basically created a large ash, water, mud mixture called a lahar. And that lahar, so another type of debris flow, flowed down the side of the mountain and completely destroyed a forest, but also impregnated that wood in the forest with this silica mud mixture. And so for about the past 15 years, scientists, or 20 years, scientists have been going out and looking at this old forest that is now becoming permineralized wood in nature as it's forming. And so we can actually gain a lot of insight into how fossils form by going out into nature itself and looking at current fossils, or sub-fossils, if you say. And this is one technique that I will employ in my next section, which I was looking at the preservation of land crabs in the Bahamas. And so land crabs, if you've been anywhere in the tropics, they're very, very widespread, they're very common. I was in the Bahamas, and two of the species that are very common on the island are these two J-carcinid crabs, J-carcinus lateralis and J-carcinus roricola. There are thousands and thousands of them. They're everywhere. They even come into your bedroom and climb on your ceiling and hang above you while you're trying to sleep. That's how common they are. And they're very, very important in island ecology. And so if you go back to that cycling figure at the beginning, they're kind of the rats of a lot of tropical ecosystems. So this island in the Bahamas has no native mammals. And so these were the guys who were eating all of the debris. These are the animals that were eating the small spiders, the plant matter, and recycling all of that organic matter back into the biosphere. But they also themselves are eaten and return these nutrients to high up in the food chain. So they're very, very important in cycling. Today, in today's ecosystems, they're very widespread. But very little is known about land crab history. When they evolved, how many species were there in the past? Were they always so abundant? Were they always so important to ecology? And so one of the things we want to do is look at land crab fossils. They're very, very rare. There's been 2 reports published ever on land crab fossils, only on this species, only from Jamaica, and only from about 2.5 million years ago. And in both examples, they only found half of a claw. So if you think of a crab claw, if you've ever been to or eaten a crab, you have the big stable part, and that's like that part here. But then you also have like the little movable finger. And so both of the fossils are just that little movable finger part of the claw. And so this raises a couple of questions. Why aren't there land crab fossils? Does this mean that they weren't around and these are a modern, is it a recent evolution? So even though they're very important to ecology today, is that only just a recent phenomena? And so there's 2 hypotheses. First, they were in the past, but they're not being preserved, they're not becoming fossils. But the second is that they evolved recently. And I decided to test this first hypothesis, that not preservation, the lack of preservation is preventing them from becoming fossils. So what I did is I went down to the Bahamas for 5 weeks, and I essentially walked around and collected dead crabs for 4 weeks, identified the species, I measured them, identified the body part. So was I picking up just a leg, was I picking up a body, or was I picking up a claw? And then I also looked at subfossils, so I went out to a few different localities in the same areas and dug up sediments and tried to find subfossil crabs. And in the end, I collected about 1,400 dead crabs, and I'll talk about what they show. So this is where I was. This is the island I was on. It's called San Salvador. It's very, very far out on the Bahamian shelf, so it's pretty removed from any sort of larger landmass. And as I said, there are thousands and thousands and thousands, maybe millions of land crabs. And this is the northern end of the island where I was doing my research. And I collected them from 3 different localities. So first, I walked along this little outcrop here. So these are a series of cliffs. I walked along this beach, and then I collected them from what used to be a catchment for water when the Geraas Research Center was a Navy base. It's now kind of overgrown. There's crab burrows all throughout it. It's overgrown with vegetation. I guess I can show you pictures of it. This is the beach where I was walking along. So I basically, from where the water met the sand to about 30 meters back from the sand, so up into the dunes, I was collecting these dead crabs. I was walking on top of these cliffs. And this is one of the areas here that I collected the sediments from. And then this is the catchment. And I was walking around and picking dead crabs up from the entire area. So what did I find? I wanted to see if, we know that the fossils are rare, but they're all claws. So I wanted to see if what was happening is that claws were being somehow preserved at least a little better than the rest of the body. So if you can think about a crab, like this one, there's 8 legs, 2 claws, and a body. So this is the ideal distribution if every crab that was dying was being completely, or every single body part was being put into the system. So this is the distribution. So you have 2 claws, 8 legs, and 1 body. If you compare this to the collection that I did of these dead crabs, in every single instance, there's more claws than there should have been based on the amount of things I found. So fewer legs, so the legs are being recycled more quickly. On the top of the cliffs and in that back area, there's way fewer bodies. There's a lot of legs and a lot of claws for almost no bodies. Found a few more on the beach. And so even within the 4 weeks I was there, and I was collecting them every day, so even within a day, things are already being recycled back into the biosphere very, very rapidly. I said that they were important because they recycled organic matter, but they're also important food sources for birds. So birds are coming around and eating the crabs themselves. A lot of times I saw ants swarming over the bodies. So they're rapidly being returned to the biosphere. So what happened when I tried to find fossil crabs? This is one of the outcrops. Kind of see this is a land snail. These are land snails. This is a marine snail. This is a marine bivalve. So I collected about 8 gallons of sediment and put it through a sieve very, very gently, and I resulted with about 2,500 different fossil shells. In those 2,500 fossil shells, I found a single claw, and this is what it looked like. No organics left, so the original claws on these crabs are bright purple. You can see that the calcite within the claw, the mineral within the claw that makes it so robust, is already starting to break down. You can see the 2 different layers there. It's very fragile. It survived long enough for me to take this picture and then just completely disintegrated, so it's no longer with us. But out of the 2,500 fossil or subfossil shells, only 1 crab claw. So what's going on? You have a huge population. You have thousands and thousands of crabs. Their death and as they, their remains are being rapidly recycled. So you have fewer remains to even be buried, to even attempt to become fossils. And as the result, and the end result, is that there are very, very few land crab fossils. So this supports the idea that preservation, or the lack of preservation, is preventing us from looking at these in the fossil record. They're being so rapidly recycled into the biosphere, into the lithosphere, that there just are no fossils. That supports the hypothesis. It's kind of a sad result. There are no land crabs in the fall. There probably were land crabs in the past, hopefully, but it's sad. So, complete change of thought. I'm going to be talking about fossil leaves from New Caledonia now. Before I talk about the fossils themselves, I want to talk about the whole plant fossil record. And the association between fossil leaves and iron is very, very striking. I went through every single drawer in the paleobotany collections and looked at about 150,000 fossils, and I found that almost every single one that had a leaf, not a compression fossil, but an impression fossil, has iron associated with it. So just like these, iron rapidly coats or iron oxides coat the surface of the leaf as it's decaying. When it's buried, the organics will go away, but you're left with these rust-colored impressions, and that's very, very common in the fossil leaf record. However, all of these, as I said, were impressions. Very few of these, this one only, have any sort of extra morphological detail. So you can see the general shape of the leaf, you can see the veins, but if you went and put this, I did look at this leaf under the scanning electron microscope, you can't see stomata. You can't see any of the details that would have been on the surface of the leaf, or on the interior of the leaf. So in none of these examples can you see that vascular tissue like you would in permineralized wood. That's all gone because they're just impressions. My leaves are different. This is the island I went to. It's New Caledonia. So this is Australia, this is New Zealand, so it's in the South Pacific, pretty far removed from both Australia and New Zealand. It broke off from Gondwana pretty early in the Cretaceous, so it's been separate from the mainland for probably 100 million years. And in terms of the modern flora, it's one of the most diverse groups of plants in the world, and so a lot of botanists go there to study, but until these, there was no paleobotanical record. And so we went to the island last April to try to find these fossils. And we collected them from these pink regions down here. And I should say that this green area here is built on something called an ophiolite, which is basically marine rocks and marine basalts that have been uplifted and pushed on top of other material, and so it's something ultramafic. It has a lot of iron and a lot of magnesium. As these rocks are weathering away, the iron doesn't really weather down. So if you think of a soil profile, if you've ever been in the tropics, you see vast red sediments, and that's because all the other minerals, all the other ions, the elements are being leached out as water flows through those rocks. The water doesn't really touch the iron, it's left on the surface. And other processes can then weather it, but these red areas down here are basically built of iron clay. That's what it looked like. This is my colleague, Andrew Leslie. He was very, very silly in that he wore white clothes on the first day. They're no longer white. My own pants, I started out with blue pants, they're now red. My shoes are red. They'll always be red because there's just so much iron in these rocks. And this is the outcrop where we collected them from. Little seam here is the bed that we found these fossils in. So what's happening here is that most of this is just soft clay. We could dig it away with our own hands. But it's this, for whatever reason, this one layer lithified. It became rock. It's very hard, very rapidly. And so we were collecting our fossils from here. And this is what they look like. I have a couple examples up here at the front if you would like to see them. They're preserved in 3 dimensions, which is very unusual for a leaf. Leaves are almost 2-dimensional to begin with, so the fact that they have any sort of dimension is stunning. And they're preserved kind of as a leaf mat, so leaf, leaf, leaf. They're not nice individual specimens. The rock itself is composed of about 70% hematite. So it's iron oxide, it's very dense, it's very heavy, it's very hard. And it doesn't break nicely, so unlike some of these fossils up here, you can't split it, it's not flat. So they're a little difficult to work with. But the leaves themselves are absolutely beautiful. So this is one of the examples here, and these are scanning electron microscope images of the details that you can see on these. So you do have stomata, you have vascular tissue. This bundle of stuff here is the mesophyll. This is where most of the photosynthesis occurs in the leaf, and it's this tissue that rapidly decays away. So if you even in the fall when you're out collecting leaves, I collected a few last fall and put them on the scanning electron microscope. That tissue was already gone after a few days of sitting outside. So the fact that it's preserved is magnificent. It means that this fossil became a fossil more quickly than it took for the mesophyll to disintegrate or decay away. You have individual cells of the vascular tissue preserved in iron oxides. Well, I guess I ruined part of my story there by saying that. But again, this is the leaf split open, so these little ridges here are where those cells were together before I ripped them apart. This is inside the leaf, looking down through the stomata. Stomata are great because they're how the plant controls its interaction with the atmosphere. So CO2, oxygen, water, it's all controlled by these stomata. And they have very, very specific climatic significance. And so if you wanted to, or if I wanted to, I could go back and count the ratio between these stomata and the other cells in this leaf and actually get a climate signal out of them. So the fact that we have stomata in these leaves is very important for looking at past climate. It's very important in that aspect. We have details of vascular tissue that I've only ever seen except for things like mineralized wood, in one other locality. So I want to draw your attention to these spirally looking things. Those are the xylem cells of the plant. And so in living plants, some of the xylem, in order to support all this water flow, has areas of the cell that are more heavily lignified. So lignin and cellulose are the 2 primary components in most plants, and lignin is what makes wood so robust. And so in these individual cells, there are certain areas that are more woody or stronger than the rest of the cell to help keep them open for this water flow. And those are preserved in these fossils, which is just, when I saw, I almost cried when I saw this. I was so happy. And the fact that they're in 3 dimensions is very cool. And the other cool part about this is this also indicates how rapidly they formed. Cellulose decays away more quickly than lignin. So in this case, The cellulose has decayed away, but the lignin hasn't. And so you can kind of figure out a time frame of when this would occur, because part of the cell is gone, but not all of it. And so by looking at, by that, we can kind of get an estimate on how quickly they formed. This is a leaf plant. So some leaves secrete various things on their leaves. Some are involved in symbiosis. So if a leaf secretes something nice and sweet for mites to eat, those mites will then clean the surface of the leaf. They will prevent other arthropods from eating the leaf. So leaf glands are really important in plant ecology. I can't find another reference to a fossil leaf gland in the literature. So this is very rare in the fact that it's one, three-dimensional. The cells around it are preserved. You can see individual cells, that's a cell, that's a cell. And then the cells within the leaf gland themselves are preserved. I did cry when I saw this. I didn't almost cry, I was very happy. And the entire leaf surface is covered with them. So it's not just this one area, it's the entire leaf, and it's spectacular. There's a few different textures in these leaves that do give some indication of how quickly they formed. So these are these large acicula. I took the scale bar out of this one. That was silly of me. I had scale bars in the other one. The frame here is about 50 microns, or 51 thousandths of a millimeter in size between this entire thing. And so each one of these crystals is about 5 microns long, 5 thousandths of a millimeter. And so these, in order to get that big, probably had more time to grow. We also see a similar texture, but much, much smaller. So this scale bar here is 2 thousandths of a millimeter. And so these are same kind of acicular needle-like shapes. They're very, very tiny. So these probably grew, the individual crystals grew quickly, but this entire thing, these formed much more rapidly because they filled in the space, and so they couldn't grow any longer, whereas these had a longer time to grow, a longer time to get bigger, a longer time to fill the space. And then there's textures. This is a scale bar. This is 1/1000th of a millimeter, and it's a texture. This is part of the stomata here. The texture is so fine that you can't even really see the crystals themselves. They're nanoscopic. And that's what gives us this great detail on this stomata. So what are these? What are these minerals that are forming? In all three instances, it gives the exact same elemental signature of iron and oxygen. Iron oxides, one of the minerals that takes that needle-like shape is called goethite, named after Goethe. I don't know why, but that's what it is. And so this is probably this iron, this oxygen, and combined with that the mineralogy, the shape of that mineral, probably indicates that it is goethite. So great, we know they're three-dimensional, they're beautiful, they're preserved in iron oxides. That's nice, but what about modern leaves? Can we get any information about what's going on in the modern ecosystem to help us understand how they're forming? So when we were in New Caledonia, we went to this little stream. You can see all of the dry rocks around it are rust-colored. The sediments themselves are red. This is one of the reasons that I had to leave my rain boots there, because they turned red. There's just so much iron. And so what we did is we looked at this stream and we looked at leaves that were accumulating in these sediments today. And these are just a couple of examples. I tried to show a variety of leaves. So this one is where I actually cut it. So that was actually a whole leaf before I took the picture. So it's completely coated in these rusty iron oxides. Some leaves have decayed away, but you can see the entire, all of the veins. So there's 5 orders of veins in this leaf, and in certain areas they're still preserved, and you still get some of this tissue. Some of this mesophyll is still within this leaf. This leaf has a little patch open, but otherwise it's intact. And so I tried to look at these to see if we could learn anything from what's going on in the modern environment. And these leaves had probably been in the stream no longer than 2 weeks. We were there just after the rainy season. The stream had gone down. We were collecting these from sediments that were exposed but still wet. And so that probably was around 2 weeks in this iron-rich sediment. And what we found, or what I found, is that these leaves were coated in this microorganism called a diatom. And so these are a type of green algae. They're photosynthetic, and they build these lovely, beautiful little skeletons out of silica. And they were everywhere on the surface of the leaf. And what diatoms do is they produce extracellular secretions. They create what's called a biofilm. And that biofilm rapidly attracts iron oxides. So on all the parts of the leaf that we saw these diatoms, we couldn't see their secretions anymore, but we found these iron oxides completely coating the surface of the leaf. And so again, these are stomata, these are iron oxides, and they're just everywhere. But because we had exposed leaves, we had leaves where the external part, the cuticle of the leaf was gone, we could actually look on the inside. And you can see here, these are some veins of one of the leaves, and they're already being replaced by iron oxides. Just by sitting in the stream for a couple of weeks, being coated by these diatoms, the iron oxides are already replacing these tissues. And so, just like we expected from the fact that we have these cellular level details, we have the mesophyll preserved, this process is occurring very, very rapidly. And yes, it's iron oxides. This is just a yes. So we can actually create a model of how these leaves formed and are forming today. So you have your plant. As they shed their leaves, their leaves are accumulating in the stream. They're being coated by diatoms, they're being coated by bacteria, which then attracts iron oxides to the surface and internal part of the leaf. And as those leaves are then buried in sediments, they're not being returned to the biosphere. Over time, in a very short amount of time, you get the resulting fossil, which then we go and collect. So I want to kind of put it back in the bigger picture a little bit. So this is another diagram showing you have this giant biosphere, you have billions of organisms, but through each one of these kind of tiers on this pyramid here, things are being removed. And so at each step, we call these taphonomic filters. At each one of these filters is removing these organisms from even potentially becoming fossils. So at this stage, you have all of your fossils. At this bottom section of the pyramid, you have the fossils that are actually collected. So there are millions of fossils out in the world that paleontologists probably won't collect. Peabody has about 6 million fossils here. That's a huge amount. There's a huge number. And that's just the tip of what is out there in the world. And why is this important? Why are museums important? Why do we have the Peabody? Why are all of us working at it? The research part of museums is incredibly important. So this is the invertebrate paleontology collections. This is a drawer of insects from there. It provides a wonderful resource for scientists to come to study the fossils, to learn new things about past life. So this is a recent paper on a 450-million-year-old organism called an ostracod. It's a little arthropod that floats around in the ocean. We have some of the Peabody that have eggs preserved inside. So we can actually study how these organisms 450 million years ago were reproducing. And that's incredible. And without these collections, that just wouldn't be possible. But it also connects science to the public. So I know that one of my favorite things here at the Peabody, the thing that I enjoy the most, is interacting with the public and teaching them about fossils. I study them because I think they're cool, and I want other people to think they're cool. And getting kids excited about science, I teach rocks and minerals to first and second graders, and getting them excited about a rock is just fantastic. And museums provide this opportunity, and so volunteering is obviously important. These are 3 of the volunteers I know best. These are volunteers in the invertebrate paleontology collections. Cope is here somewhere. He'll give me a hard time later. And I want to thank all of you, and I know that I think we all put in a lot of effort for the Peabody just because it is an important resource for everyone. And so thank you very much. And I have to thank some people and some funding sources and the fossils. Thank you.
I know from personal experience, it took about 70 years to become a living fossil.
Yes.
The question that I have, can you tell us a little bit about animal fossils and the time period involved, for example, dinosaurs?
So how long would it take for them to become a fossil? So I guess we have to go back to, there's two types of fossils. You have the big skeletons that we have in the Great Hall, where we just have the bones. And so for those, the big issue for a dinosaur to become a fossil is that they're huge. If they're dying on a hill somewhere, the time the most important part about that is transport. So how long does it take for the bone to be deposited in either a river or a lake? The most bony fossils, a lot of shelly fossils as well, are actually permineralized. So if you've ever held a modern bone and a fossil bone, the fossil bone is much heavier. The void spaces within the bone have been permineralized. There's often like calcium carbonate or silica that's added to all those spaces. That can take, that process, once the bone is buried, millions of years. Millions of years. It's usually a very, very slow process. So silica, silica in wood can be deposited very, very quickly because silica has an affinity, it really likes cellulose, and so it will bond very quickly. But even in permineralized wood, initially the silica deposition is very, very rapid, and so the direct surface of the cells will become permineralized very quickly. But then that remaining void space can take thousands, millions of years for all of the space to be filled. And bones, same thing. So for really the bones, millions of years. For things like the feathers, like that Confucius ornis I showed, I would say that once it's buried in that volcanic ash, I'd probably call it a fossil. But even those bones have been altered, they've been changed over the past 80 million years.
Thank you.
I'm going to go here and then Francis.
Yes.
I wanted to go over some of the terms you use.
Sure.
The difference between compression
Yes.
and impression. And the second question is, all those leaves you found in Caledonia, how were you able to figure out what the climate was like during the time that those fossils were formed?
The tricky thing that I left out in the New Caledonia fossils, I'll touch on this first, is that in terms of how old they are, that's something that our group is still really trying to, I'm just trying to go back to the slides, sorry. That's something that our group is really trying to work on today. Our best estimate for the age is 5 million years, plus or minus 5 million years. Really precisely. In reference, a lot of fossil localities can be dated down to thousands of years from millions of years ago. So it's a good date for a fossil might be like 65 million years, plus or minus like 0.1 million years. That would be great. In some areas, it can be even more precise. And so I want to preface that by saying we don't know how old they are, and so we can't really say anything about the climate because we could, but we would have no idea when that climate was. In terms of the climatic analysis, we collected thousands of fossils. They're still being identified. And so like a lot of, like I talked about getting climate out of stomata. None of that work has been done. That being said, there are general broad associations you can make with different leaf types. So if you have a fossil assemblage with lots of large leaves, that meant, one, there was enough water for a tree to support those large leaves, but also probably it was in the forest. You need a lot of area on your leaf to have more photosynthetic area. All of the fossils we collected are pretty small leaves. Like the largest fossil leaf we found probably 2 inches long or so. So this is probably a drier area. It probably matches a lot of the flora of the southern half of New Caledonia. It's called, what's it called in French? I don't know. It's called steppe vegetation. So it's dry, it's adapted to seasonal availability of water. So New Caledonia has a very, very strong dry and wet season. So it loves water, but it has to be able to survive for 6 months of the year without it. And so that's why a lot of these leaves are small. So we can say broad things like it was probably in a seasonal environment. It's probably hot because New Caledonia has been in the tropics for the past 80 million years. So the tropics are always warm. But that's about it. In terms of compression, impression, now I'll turn off the light so you can see the bee because he's my favorite. Compression just means that literally the leaves fell into sediment, or this bee fell into sediment and were compressed. And so you can think of it, it's harder to think about in terms of the bee. It was originally 3 dimensions. You had this bee. It fell down into the water, landed in this position, and was just squished down. And so you get all of the anatomy in this basically film on the rock. Leaves are already fairly 2-dimensional, so the compression doesn't do much. Difference between compression and impression is that a compressed fossil, a compression fossil, has carbon left, has some of the original tissue left. It's no longer chemically the same as it was, but it's still organic. An impression is just like a leaf falling into cement, leaving its impression in the cement, but there's no tissue remaining anymore.
From
I have 2 questions.
Sure.
The first one is, this is in the case of the dinosaurs, when they die, how long does it take for the soft tissue
Oh
to
yeah.
regenerate?
Well, it depends on where it's buried. It depends on who's around. Ideally, let's say, it was buried by an ash cloud. So excluding scavenging. Muscle tissue can begin to break down within, any of you have seen rotten meat? A couple of days, a couple of weeks. Generally, muscles are kind of the first thing to break down. Additionally, one thing about bodies is that we have a digestive tract with lovely bacteria that love to break things down. And as soon as we die and are no longer able to control that, those bacteria also break down your body. And so that's one of the things that in order for soft tissues to be preserved in animals, like those Burgess shale fossils I showed earlier, you kind of have to